A very good day, my dear undergraduate and postgraduate students. The topic for the day, tumor suppressor gene, is specific for the postgraduates, though it is not uncommon to find such a question in the undergraduate question paper also. Hence, specifically the postgraduate students, I would like you people to kindly, deliberately follow each and every word that I say. This is a very abstract, difficult topic, particularly at the exam time. We shall go to the topic proper. Self-expression must pass into communication for its fulfillment. There were two terminologies which I was interested in this context, and one was expression. Obviously, you find that there has to be an expression of a property. Perlesbeck, again, the wonderful author of Good Health. The subheadings for today shall be all this. What is the tumor suppressor gene? What is the list? And these particular genes, RB, P10, P53, Hedgehog, etc. What is a Knutson's hypothesis? What is an APCG? The diagrams. And this one happened to be the major reference or the backbone of my presentation. I would be very much happy if you people can go to this reference and read it thoroughly. Always pictures first, text next. So let us see what the picture says. In this, there is a normal cell, and then there is a tumor cell. So when there is a fusion of the normal and the tumor cell, ultimately you find that there is a hybrid cell that is produced, which is non-tumorigenic. In fact, each one of us has got an abnormal chromosome and a normal set of chromosomes. But still, the abnormal property is not to the front. This is because of the suppression of tumorigenicity by the cell fusion. The fusion of the tumor cells with the normal cells yields hybrids that contain both normal as well as the abnormal chromosomes from the parents. And these are non-tumorigenic. That is what is shown over here. This was initially attempted to be expressed by Henry Harris in the year 1969. But then he did not have a perfect justification to his finding. And later on, you find that there was another set of body that developed it. Hence, something in the normal cell is preventing the growth of the tumor. So that is the tumor suppressor gene. Something is preventing the growth of the tumor, which is called the tumor suppressor gene. Tumor suppressor genes have been identified in tumors, but in that case, they themselves are defective, so the tumor is progressing. First, we shall go to the interesting RB gene or the retinoblastoma gene. So, this was the first tumor suppressor gene that was identified. If identified early, retinoblastoma can be treated well. That is what the reference says. Some of them are inherited as Mendelian dominant. As I told you, we'll go to the picture first. Here is a female who has got a defective chromosome, a male with a normal chromosome. And when they give rise to the offsprings, you find that there can be a defective and another defective one. 50% of the offsprings are affected, whereas the other 50 are not. Now again, what will happen is when there is going to be a fusion between this, there can be another set of abnormal chromosomes. So as a result of which, one, it can be a direct inheritance, or there can be a mutation that is developing, which can result in a tumor. If that is not so, the individual continues to be normal. So it is the second hit that matters, which is the Knudsen's hypothesis. This gives rise to a defect in the RBC tumor suppressor gene, which results in retinoblastoma. Look at this one. What is this defect? It occurs in the 13q14. Q is the long arm, and at the segment 14 it occurs. So this is the retinoblastoma gene, and when there is a deletion of it, you find that there is a retinoblastoma. This is actually a tumor suppressor gene. When it becomes defective, it can lead to a retinoblastoma, but luckily we are being protected by the other chromosomes. RB gene is normally active in all the individuals, including you and me. It is inactivated by phosphorylation. RB gene cell cycle clock is removed by the loss of heterozygosity. So, one from the mother, another from the father, they are heterozygous. But when this heterozygosity is lost and it becomes a homozygous defect, then there is a manipulation of a tumor due to deletion of one or both the chromosomes, or the normal copy that is being there is also mutated. So this is a wonderful explanation regarding Knudsen's hypothesis. If you are not convinced, see this picture again. A superb picture thanks to, I do not know how to spell this person's name. Pesner probably, W being silent. So normal chromosomes, maternal and paternal, both are having the tumor suppressor genes. So nothing wrong. But first hit, it can be genetic also. There is a mutation or an inheritance. One chromosome is defective. This is the first hit. Again, nothing will happen to the individual. But sometimes there can be a wild type of chromosome. There can be a chromosomal loss or a mutation whatsoever. So this also becomes defective, or this is being lost. As a result of which, there is no suppressor gene that is there. There is a manifestation of neoplasm. This is the wonderful two-hit hypothesis of Knudsen. I would like you people to kindly remember and draw this diagram. If you are not very convinced, see this again. As I told you, pictures are pictures. Thanks to New York Times for this reference. So non-hereditary, a normal cell, so both the chromosomes are normal. One hit, there is an abnormality or a mutation in one of the chromosomes. But then rarely there can be a second hit, as a result of which you get a retinoblastoma gene that is developing. The patient can develop a malignancy. In another one, the normal ones are not being heredited, and you find that there is a defective chromosome. And again, in the event of a second hit, there can be the development of the retinoblastoma gene, which means the tumor suppression is lost, the tumor manifests. And this is our wonderful person, Knudsen. Obviously, he has been given the Nobel Prize. Loss of the RB gene can lead to retinoblastoma. It can lead to other tumors also, lung, breast, osteosarcoma. If you go to your text on osteosarcoma, the RB gene will be one of the causes. Also, there can be infections with SV40 or the human papillomavirus, which can result in osteosarcoma and mesothelioma. So SV40 infection is there, and there is the inactivation of the RB protein by the T antigen. There is a transformation of the cell into a malignancy. A wonderful set of explanation, and all the credit gives to the reference that I had quoted first. Next is the P53. This also is a university question. Retinoblastoma gene is also a university question. This is also a tumor suppressor gene. And when the tumor suppressor gene is activated, you can get leukemias, lymphomas, sarcomas, and brain tumors. Carcinoma of the breast, lung, and colon. 50% of the cancers, they say, are related to P53. And it is the most common genetic target. Rarely it can be a hereditary transmission, in which case the same patient can be presenting with multiple malignancies. Probably xeroderma pigmentosum. There is a defect in the telomerase also in that therein there can be multiple malignancies. The target oncogene protein can come from SV40 or human papillomavirus as in retinoblastoma. So the activation of it, the tumor suppressor gene, the proto-oncogene becomes an oncogene. This is the basic principle that I had told you people in the first class. So it can be because of the mutation of the proto-oncogene. Normally the proto-oncogene is not harmful, but it can become harmful because of the loss of regulation or a misregulation. Two things. Or there can be a chromosomal translocation, relocation, and it is better expressed. Therefore, there is a malignant transformation, becomes an oncogene, and probably there is a hybrid protein, the stem cells leading to a leukemia. This we shall be having in one corner of our mind. Now, how does the P53 function? Look at this one. This is a DNA that is being damaged, and this leads to an increase in the level of the P53. The P53 normally, it is supposed to be a protective one, and it protects this, as a result of which you find that there is the continuous production of the P21, as well as it leads to the formation of CDK and PCNK, proliferating cell nuclear antigen. So these two are being produced. And what happens is they lead to inhibition of the cell cycle and the inhibition in the DNA replication. So the cell cycle is also stopped, the replication is also stopped, as a result of which there is no malignancy. Normally, you find that malignancy is being triggered by a defect or an injury to the DNA. But what will happen if the P53 itself is inactivated due to some kind of a mutation or a damage? Then you find that this control is being lost, as a result of which you find that there is no inhibition of the cell cycle by the CDK, and the PCNA DNA replication is also not stopped. As a result of which you find that there can be a continuous production of defective DNA leading to a neoplasm. This is yet another one. The damaged DNA is here, leading to the activation of the P53, and there is the cell cycle arrest usually or the apoptosis. But when there is going to be a defect in the P53 or an inactivation of it, there is a continuation of the cell cycle and there is no apoptosis. Uncontrolled proliferation takes place. This is what is given in this text. So loss of the P53 will lead to induced cell cycle, as a result of which there is increased mutation, there is increased instability, and genetic instability is a common property of all tumor cells. It can contribute to further alterations in the oncogenes. The action of P53, the wild cell type again is, it is required for both cell cycle arrest and apoptosis. But when this is going to be defective, you find that the cell cycle and the uncontrolled proliferation continues. Next one is the APC gene. The APC gene is adenomatous polyposis coli gene that has been identified by the Johns Hopkins Hospital. So it inhibits the mutation by complexing with the B catenin. And sometimes there can be a mutation in the APC, or there can be a mutation in the B catenin, or there can be a mutation in both, as a result of which you find Once there is a mutation of the regulator gene, you find that there is an uncontrolled proliferation. So APC, it becomes inactivated. There is a degradation of the B-catenin also. And the B-catenin, it enters the nucleus and activates the MYC and the cyclin 1, etc. As a result of which there is a cell proliferation. So whenever there is going to be a defect in the APC or the defect in the B-catenin or in this complex, there can be a cell proliferation. Remember it like this. Whenever there is a promoter, then the activity of it will lead to cancer. But whenever there is going to be an inhibitor, then the defect in the inhibitor will lead to a cancer. So that's the germline mutation. There can be mutation in the APC. This one box, if you people can remember, it is more than enough. Mutation in the APC gene, mutation in the B-catenin, mutation in the APC B-catenin pathway, which can lead to cancer. Cancer need not be only the colonic carcinoma, but there can be hepatocellular carcinoma, hepatoblastoma also. So where mutated APC alone, it can lead to familial polyposis. On second mutation, due to various causes, including viral infections, there can be a carcinoma. This is again a wonderful diagram which you people can use for many analyses. There is an APC over here. This is a normal epithelium that is there. What happened? This is a chromosomal instability pathway. So there is an APC loss, there is an early adenoma, then there can be the other oncogenes such as the KRAS and so on. And what will happen is they lead to a late adenoma. Again, P53 inactivation, our friend in the previous discussion, all will lead to a cancer. This is a wonderful picture and I would like you people to kindly have it in your mind. You can draw it for many analyses. Microsatellite instability is a question for you. Again, in this case, there is a APC loss. It's the same through failure of the MMR genes. And there can be mutations of the TGF, BAX and others. And therefore, an adenoma that has been produced becomes a cancer. The next one will be the serrated pathway. Serrated lesions of the intestine is a recent advanced question which I have covered in detail. I would like you people to kindly go back. So in this, there is a BRAF mutation or a DNA methylation. There is a serrated adenoma. Again, inactivation of the P53, etc., P16, and methylation or mutation of the genes can lead to a cancer. So these are the various pathways, and you should understand that the APC is the colonic cancer gatekeeper. It prevents it from entering. Thanks to ResearchGate for this wonderful diagram. A superb picture, definitely the best part of this presentation. 3 answers in one diagram. Now, what is the gene? What is the type of cancer? Of course, you will not be having a MCQ. You have passed that stage. But then still, if you are able to rattle off in your Viva, etc., definitely you are going to score the additional marks. And if the tumor suppressor gene is going to come as a question, definitely I would like you people to kindly mention this. And then the type of cancer. P10, for example, in brain tumors, prostate, endometrium, etc. PTC in basal cell carcinoma. NF1 in neurofibrosarcoma. And WT1 in Wilms tumor. So the entire list is there. BRCA1 and 2, you people know breast malignancies. P10 is the next tumor suppressor gene. So this is the normal function over here. There is a P1 kinase and there is a diphosphate which is getting converted into phenoacetyl triphosphate and it combines with AKT and there is a cell survival over here. Now what will happen is this is being prevented by means of this P10. P10 is a tumor suppressor gene. So it is a lipid phosphatase that dephosphorylates PIP3. So this is not over here and therefore this is not formed. This complex is not being formed, as a result of which there is a delayed cell survival and there is no malignancy. But what will happen is when this P10 is going to become defective. So this is a tumor suppressor gene. When this becomes defective, then this process is not stopped. PIP couples with AKT, as a result of which there is an increased amount of cell survival, which will lead to malignancy. Hedgehog polypeptide is another one. This is a hedgehog. I do not know why that name was given. It gets attached to the cell surface. And normally you find there can be this one. There can be the activation of the microtubules and cytosol, etc. And finally, the nucleus. It helps in the formation of a transcription leading to a malignancy. So the rest of it is given over here. It disrupts this kind of a margin, coastal margin is being disrupted. Therefore, there is a cycle that leads to malignancy. This is the hedgehog polypeptide, probably because of the serrated surface or whatever it is called so. What are the functions of the tumor suppressor genes? One, they are opposites of the proto-oncogenes and oncogenes. So these two are helpful in the formation of malignancy. Tumor suppressor genes suppress the malignancy. Proteins encoded by this inhibit the cell proliferation and survival. There is inactivation of this tumor suppressor gene, it can lead to tumor. And that can be because of these mechanisms. Elimination of a negative regulatory protein or TSG, tumor suppressor gene function, is opposite to that of an oncogene. Therefore, you find that when this is removed, oncogene becomes active, there is a neoplasm. People tend to suppress that which they cannot express. A beautiful quote. I do not know what it means. We tend to suppress something that we cannot express. I hope this class has conveyed something to you people which I have found difficult as a student. Tumor suppression and gene expression are the important components of today's class. Thank you.